Sorry, that should have been muted.
It's perfect.
Hey,
This is the
Elena.
excitement of 116 people.
You're making me more nervous.
Jen, you're muted if you're saying anything. I think the live streaming is working now, right? You got it.
Sorry, I was trying to log into the wrong email, y'all. Okay, so this is first time back, jitters, apologies. Okay, we're just going to go ahead and get started. So sorry for the delay. That was 100% my fault. So hello, everybody. Welcome back to Cell Migration Seminars. So this is our, I don't know, 7th season, something like that. I think Kim is our 69th or 70th presenter. So we're so excited to be back and to be doing this again. And so just to introduce ourselves to anybody who's new to this. So this online series started in May of 2020, basically when the pandemic first hit and things were uncertain and we knew all of our favorite cell migration meetings were going to be canceled. We decided to do this online meeting, and then it's really just continued to be a great community and a great place for people to share their work and to connect. And so we've continued it, and we do hope to continue it in the future. And so this is just my plug for everyone to keep coming and to keep asking your great questions. I think one of the reasons that it's still fun for the speakers is that all of you in the audience are such great participants, and so they're not just talking to an empty room. So I do hope that we can keep this going. So just to introduce myself and the other co-hosts, so I'm Jen Mitchell. I'm a relatively new professor of biology at Wesleyan University in Connecticut, and my co-host is Dr. Ankita Jha, who is a postdoc in Claire Waterman's lab at the NIH. And I study collective migration of epithelial cells, and Ankita studies a lot of different things, but these days mostly focusing on blebbing in single cell migration. Okay, so enough about us and enough about the seminar. So we're really, really excited to have our first speaker of the series, and we'll be going through, I think, November 29th. So every week, same Zoom link. If you've already got the Zoom link, just go back to the same one, same time, 11 a.m. Eastern time. So when the time zones change, just stay with the Eastern time. And okay, I'm going to stop. Ankita, go ahead.
It's amazing to see everybody again, and really grateful that our session is starting with Kimberly Stroka. Professor Kimberly Stroka is currently Associate Professor in Fischl Department of Bioengineering at University of Maryland, College Park. Her lab generally explores the role of mechanical forces in physiology and pathological phenomena, including cell migration and cell barrier function. I think today she is going to mostly talk about cell migration in confined environment. Professor Stroka received her PhD in bioengineering from the University of Maryland, where she came back, I guess, and she completed her postdoc training at Johns Hopkins University in Institute of Nanobiotechnology. And her postdoc work also focused on microfabrication, molecular biology, live cell imaging, and uncovering mechanisms of metastatic tumor cell migration through confined microenvironments. So I would really like to welcome Dr. Kimberly Stroka, and I will give the stage to you. Thank you for joining us.
Absolutely. Thank you so much for the opportunity to give this talk. I'm really excited. Thanks to Jen and Ankita for the invitation. It's really an honor to be here. And thanks to everyone who's joining either Zoom or on YouTube. So yeah, today I'm going to be talking about some of my lab's work exploring mechanisms of cell migration in confined microenvironments. So just to kind of give you an overview of my lab in general, so we really think about microenvironment modeling and like what types of biochemical and physical cues from the microenvironment should be considered in an engineered in vitro model, because there's a lot of different cues to choose from and we can't choose all of them at the same time. So, how do we pick and choose which environments that we can include in our in vitro models, and how do they then impact cell phenotypes in those environments? And we really think about a lot of basic cell processes like cell barrier function, migration, adhesion, division, even differentiation of cells. And these are all really important for just general cell physiology, but also in many cases, transition to pathological conditions. And then while we mostly do pretty fundamental work, we're always thinking about what's the clinical relevance? How is this information even useful from a translational standpoint? So my lab has kind of 3 areas that we focus on: vascular mechanobiology, cell migration and invasion, and then stem cell engineering. And we have projects in all areas here, but many of the projects actually overlap in multiple of these areas. And so today, I'm going to focus mostly on cell migration and invasion and touch a little bit on some of the vascular mechanobiology stuff too, just give you a couple snippets of that. So my journey in the field of cell migration actually began when I was a PhD student in Helene Maranta-Espinosa's lab. And I did do my PhD at University of Maryland, where I am currently. I ended up coming back to Maryland after my postdoc at Johns Hopkins. And it's really awesome to be back in the department. My office is actually right next to Helene's. So right back to kind of where I started with my cell migration journey. But in my very first project, looking at cell migration, I was studying neutrophil migration on polyacrylamide gels of varying stiffness, and just looking at basic things like their morphology and also their migration. I found that the migration speed was biphasic with substrate stiffness, and there were morphological changes that kind of went along with that as well. But then I started kind of thinking a little bit more about the process of neutrophil extravasation and how neutrophils actually squeeze through endothelial cells during either an immune response or even in cardiovascular disease. And so I started thinking about neutrophil transmigration. I kind of used a model where we had human umbilical vein endothelial cells on polyacrylamide gels of varying stiffness. And what we found was that neutrophils on very soft surfaces had lower levels of transmigration in comparison with neutrophils on endothelial cells that were on stiffer substrates. And so we published this work in Blood and kind of were able to uncover a mechanism where myosin light chain kinase activity in the endothelial cells on the stiff substrates kind of led to higher contractility in the cells, which allowed the neutrophils to kind of squeeze through at the junctions. And this idea has been really kind of corroborated across many different labs, this idea that endothelial cell monolayers on stiffer substrates tend to have higher amounts of actin stress fibers, leading to this higher tension at the cell-to-cell junctions, which then makes them more permeable. So yeah, this was kind of my first introduction to the field of cell migration as a PhD student. And towards the end of my PhD, I became really interested in tumor cell extravasation, which in some ways seemed similar to neutrophil transmigration, but in many ways is also different. So, when there's this process of cancer metastasis, we start out with a primary tumor. The cells can start to become invasive, intravasate into the bloodstream, kind of go through this circulation in the bloodstream, roll, arrest, and maybe stop at a location in the bloodstream, in the vasculature, extravasate, migrate through tissue again, including migration through these confined spaces, and eventually form a secondary tumor. So my lab has studied multiple aspects of this process. We've published papers in kind of multiple aspects of this process, from cell invasion, transmigration, migration and confinement, thinking about endothelial cell barrier function. And actually, a large part of my lab right now is thinking about these endothelial cell-to-cell junctions and how they change in different pathological conditions. And so in this talk, I really want to highlight a few snippets of projects related to cell migration and confinement, and then dive deeper into one particular recent project that we've worked on. So the first thing, kind of a snippet that I want to talk about, is how tumor cells squeeze through the vascular endothelium. This in some ways is tumor cell migration in confinement. It's maybe a little different than what you might expect, like cells migrating through a microchannel. But let's just kind of talk about how the cells do this. So with neutrophils first on top here, kind of going back to some of my PhD work, the process of neutrophil transmigration, as these neutrophils are squeezing through the endothelium and then they migrate underneath the cells. So in these movies, the cells are bright white when they're on top of the endothelium. And then after they transmigrate, they become darkened. And then they start migrating under the endothelium. And then on the right, we have the neutrophils in red and then the V-cadherin GFP that's labeled in the endothelial cells. And there's couple things to note here. First is that for neutrophil transmigration, this occurs over the time scale of minutes. And the total video times here are 30 minutes. And also the endothelium stays intact. So if you kind of follow one neutrophil as it's transmigrating, the junction will kind of open up a little bit, but then it closes back together after the neutrophil goes through. And sometimes other neutrophils will go through in the same spot before it closes up. But in contrast, tumor cell incorporation into the endothelium, in vitro at least, looks very different. So here we have MDA, MB231 cells, which are metastatic breast tumor cells, and they are on also on HUVECs, human umbilical vein endothelial cells. And the endothelial cells over here are also transfected with V-cadherin GFP. And in this case, we see tumor cells kind of incorporating into the endothelium. This is occurring over the timescale of hours. The total video time here is about 20 hours. And what's of note is that the endothelium becomes significantly disrupted during this process. So as opposed to the neutrophils, which squeeze through and then the endothelial cell-to-cell junctions can go right back together, the tumor cells seem to significantly disrupt the endothelial cell junctions. So we quantified different aspects of this process. And what we really found was that as the the tumor cells start to incorporate, they first kind of open this small or induce this small opening in the endothelial cell junctions, specifically in the V-cadherin, and it just kind of grows bigger and bigger. And eventually the tumor cell just kind of takes the spot in the monolayer. And the junctions around the tumor cells disappear. And even some of the junctions adjacent start to become more perpendicularly oriented, indicating that there's some disruption there. So we know that there could be multiple ways that the tumor cells could be squeezing into this endothelium. And so the first thing we wanted to look at was, are the cells secreting something that's changing the junctions or maybe disrupting the junctions or weakening the junctions a little bit to allow the tumor cells to then squeeze through? So we took conditioned media from the metastatic tumor cells and applied it to, in this case, these are human brain microvascular endothelial cells. And we observed the junctions. What we found was this transition to a very jagged junction phenotype in the cells treated with these tumor secreted factors. And so over the past couple of years, my lab has developed what's called, we call it the Junction Analyzer program, which is able to quantitatively phenotype these cell-to-cell junctions. And so we published that in other work several years ago. But what we were able to do is basically show that the tumor secreted factors contain a large amount of VEGF, that's vascular endothelial growth factor, and that if we add an anti-VEGF antibody, that actually eliminates the effects of the tumor cell secreted factors on those HBMEC junction phenotypes and essentially restores the junctions back to the state before adding those secreted factors. So kind of back to that like more linear type junction. So it seems like these cells are secreting these factors, in particular VEGF, which changes the junctions, weakens them possibly, and then maybe allows the tumor cells to kind of squeeze into the endothelium. So I kind of mentioned before this idea of sub-endothelial matrix stiffness, and that influences neutrophil transmigration. So we were also interested in how this might influence tumor cell incorporation into endothelial cell monolayers. And so we used a system of HA, this is hyaluronic acid, gelatin. We were able to tune the stiffness in a fairly narrow range by changing the concentration of extra link, which is a cross-linker in this system. And what we found was actually, sorry about that, kind of counterintuitive. On softer substrates, the cells were more spread out. They had a larger area. They were actually more elongated as well. And then on the stiffer or more highly cross-linked HA gelatin gels, they were smaller and more rounded. And a lot of times we think about like cells spreading on other types of gels like polyacrylamide gels. And normally we see that these cells are more spread out on stiffer substrates. But this is actually the opposite of what we saw with PA gels generally. Interestingly, the cells were more migratory on these less cross-linked gels, higher speed, and less migratory on the more highly cross-linked gels. And we also saw what we would have expected with these results, where their actin stress fibers were more defined on the less cross-linked gels and more diffuse on the higher cross-linked gels. So there's an interesting interplay here, I think, between the stiffness, the cross-linking density, maybe the presentation of adhesion protein, possibly porosity. I think there's a lot of factors that kind of need to be investigated. But what we wanted to do next was look at how this might change when cells are actually incorporating and migrating within a monolayer of endothelial cells. So we, again, we use cell tracker red stained MP231 cells on human brain microvascular endothelial cells. And so we looked at the cumulative incorporation of the tumor cells over time and found that there wasn't really a distinct trend in terms of the concentration of the cross-linker. But interestingly, after incorporation, so after the tumor cells had incorporated into the endothelium, and presumably they're feeling the substrate stiffness below them at this point, there again doesn't seem to be a dependence of cross-linking density on the speed of the tumor cells. And if you kind of look across the speed, the average speed over that post-incorporation time, we really didn't see the same trend that we had seen before. That was pretty distinct, where the cells were faster on the less cross-linked gels and slower on the more cross-linked gels. So that mechanosensitivity seems to go away after the tumor cells have incorporated into the endothelium. So we're kind of still thinking about this process and what that means, and cell mechanosensitivity when cells are by themselves versus when they're in a monolayer of cells. It seems to be different. And possibly the presence of those endothelial cells, which might be to some degree restricting their motion, could certainly play a role. So those were kind of just a couple snippets of what we're doing in terms of tumor cell incorporation into endothelial cell monolayers. But what I really wanted to spend the majority of time on today was talking about this process of cell migration through confined spaces, and specifically microchannel-like structures. So there was a really nice review paper that the Konstantopoulos lab put out a couple of years ago. This was actually where I did my PhD work, but this review paper was by Colin Paul, kind of summarizing what we know about cell migration in confined microenvironments. So we know that cells can migrate in these confined microenvironments in different situations in vivo, and intravital imaging that's been done, for example, by Peter Friedel's lab, has revealed that cells can encounter pores in vivo ranging from 1 to 30 microns in diameter and 100 to 600 microns in length. And these types of pores and channels can occur, for example, in bundled collagen fibers, in fibrillar collagen, along or within blood vessels, and also kind of along other types of anatomical structures like muscle fibers or epithelial surfaces along blood vessels, perhaps. So there has been a great amount of work, I think, over the past decade or so that has really tried to identify the effects of this physical confinement on cell behavior. There's been many, many labs that have really been exploring different aspects of this. And so, morphology of cells is obviously affected when cells are kind of squeezing into these confining spaces. Migration of tumor cells, cell division, the cell cycle is impacted. Even the things that cells secrete and cell differentiation can be affected by confinement. And so my lab has been thinking about tumor cells to start with, but more recently, we've also been interested in mesenchymal stem cell migration in confinement. And we're working with some collaborators to also look at the mesenchymal stem cell secretome in confinement from cells in confinement, and kind of using confinement as an engineering or manufacturing cue to give cells to improve extracellular vesicle production from MSCs. So I'm not going to talk about any of that today, but really we're focusing on the tumor cells today, and really thinking about tumor cell migration in these confined spaces. So again, this past decade or so of work has really shown that cells in confinement, a lot of things change in the cells in confinement. YAP-TAZ localization changes, indicating that mechanoactivity of the cells seems to be different. Localization of various cell organelles like the mitochondria change. Some mechanosensitive ion channels become upregulated. The actin filaments, microtubules tend to have different organizations and localizations. The focal adhesions look a little different. And different receptors are upregulated. So there's a lot of things that change in these cells. And again, there's been a lot of great work done that has identified some of these changes. So we've taken an engineering approach, and we wanted to create engineered devices, microfabricated devices that could isolate specifically the physical cue of confinement. Now, we know when cells are migrating in vivo, there's a lot of cues that they're experiencing. There's physical cues, there's biochemical cues, there's other cells that they're interacting with. But we really wanted to isolate this one specific cue of confinement. So we generated these PDMS-based microchannel devices containing microchannels of varying width. And there's a chemoattractant at the top of the device. So cells migrate in response to that chemoattractant gradient. And you can see that the morphology of the cells looks very different. Cells in the widest channels, these are 50 micron wide channels, they look very similar to 2D migration, whereas the cells in the narrowest channels have a morphology where they're completely confined. If the channel is narrow enough with a small enough cross-sectional area, the cells will take up the entire channel cross-sectional area. And for example, in the 3 micron and 6 micron channels, the nucleus also becomes deformed. So the morphology and the migration of the cells depends on the degree of confinement. And like I meant, sorry, keep going too far. As I mentioned, my lab has been interested in a lot of different aspects of cell migration and confinement. We've been using these microchannel devices to really explore the mechanisms of cell migration for both tumor cells, but also mesenchymal stem cells. And again, I'm not going to talk about this today, but we've also developed a micro pillar device, not where cells are on top of the pillars, but where cells actually go in between the pillars. And that creates a confining cue. And this is a much more high throughput system than the microchannels. The microchannels are nice for kind of exploring cell migration mechanisms, but for other work where we're more thinking about scaling up like extracellular vesicles from MSCs, for example, we need something that's higher throughput. So these microchannel or micro pillar devices have been useful for that. So again, the Konstantopoulos lab put out this really nice review paper that has this decision tree that really highlights the many decisions that cells have to make when they're choosing a migration mechanism, and also kind of highlighting the different things that impact the cell migration mechanisms. So there's a number of both intrinsic and extrinsic cues that influence the migration mode used by these confined cells. And I'm not going to walk through the entire schematic here, but things like the pore size, the degree of cell adhesion, the degree of cell contractility, a linearly elastic matrix, the cell type here. There's a lot of different factors that go into the cell migration mode. And really, it's also highlighting that cells have a lot of different migration modes at their disposal. So I'll just kind of narrow in here just for a minute on one of these. This is what we call the osmotic engine model for migration. This is something I worked on during my postdoc. And just to kind of give you an overview here, we found that when some types of tumor cells are migrating in these microchannels, they can employ what we call this osmotic engine model, where the cell can take in water at the leading edge and then expel water at the trailing edge, which leads to cell migration in the right-hand direction in this case. But we worked with Dr. Sean Sun's lab to develop a mathematical model to explain this process. And one thing I just wanted to note here is that one of the parameters here is alpha, which is a water permeation constant. And so in this equation that we ended up with for velocity, if alpha goes to 0, the velocity of the cell goes to 0. So if there's no water permeation, the cells don't move. And we had several key experimental results that agreed with our theoretical predictions based on this mathematical model. So first, knockdown of one specific isoform of aquaporin or NHE1, which is a sodium hydrogen transporter, significantly decreases migration and confinement. So up here, we knocked down aquaporin 5 and saw a significant reduction in cell velocity in the microchannels. Again, kind of in agreement with our model here. Now, there's many isoforms of aquaporins, and cells tend to express multiple of these, so there could be other ones at play at the same time. But also, there were specific cell ion channel and aquaporins that polarized to the leading edge of migrating cells in confinement, and that was also predicted by our model. And then finally, osmotic shocks at the leading or trailing edge can drive migration. So for example, here, when we hypotonically shocked the cells at the leading edge only, they reversed direction of migration and started migrating in the opposite direction. So we quantified the migration velocities of the cells, and they were in agreement with what the model predicted. Again, this was my postdoc work, but it really started getting me thinking about what's driving this process of cell polarization in terms of these aquaporins and the ion channels. And so a couple of years ago, I teamed up with Dr. Vula Milley, who is a senior investigator at the NCI. And we were funded by, this is a really cool program that Maryland has with the NCI. It's the NCI UMD Partnership for Integrated Cancer Research. So a graduate student from a lab at Maryland can kind of be co-advised by a senior or an investigator at NCI and then the Maryland lab. And the idea is that, they learn things from both labs and they split their time between the labs, and it's a really cool program. And I think the students benefit a lot from it. But we teamed up with Dr. Milley's lab to think about this question of whether RNA localization is controlled by the physical cue of confinement. And then does that have a functional relevance for cell migration? Again, just kind of going back to this idea of the polarization of cells in confinement. There's a very, it's understudied whether it's protein transport versus it could also be local mRNA translation that can be setting up this polarization of cells in confinement. So as we go on, the majority of the work here was done by Dr. Becca Moriarty. She's a lab alum of mine. She defended her PhD last summer. So we, again, have been interested in this subcellular localization of RNAs. And more specifically, we've been focusing on APC-dependent RNA. So APC is the adenomatous polyposis coli, which is, it's classified as a tumor suppressor gene. And what the Millie lab has previously shown is that this certain type of APC-dependent RNA called Rab13, this is actually a gene that encodes a GTPase that's important for vesicle-mediated membrane trafficking. It's shown to play a role in cancer progression. It's the RNA that we're going to be focusing on for the majority of this talk. So that's Rab13. So she has found that Rab13 tends to localize peripherally in tumor cells. So they use fluorescence in situ hybridization to label these RNAs. And so they've also developed a peripheral distribution index, or PDI, which is a quantitative measure of how distributed these RNAs are in the cell. So PDI of 0 means that the RNA is mostly perinuclear. If it's close to 1, it's kind of more randomly all throughout the cytoplasm. And then if it's 2, it's peripherally localized. So here they're just showing that the Rab13 is more peripherally distributed in comparison with row A, which is known to be more perinuclear. So they have also shown that these different RNAs, specifically the Rab13, tends to localize at the invasive front of migrating cells, especially collectively migrating cells. So we see them here at this invasive front of the cells. And so one way that we can assess the functional relevance of RNA localization is using these morpholino oligos. So they have developed these morpholino oligos that are directed against localization sequences in the 3' untranslated region of the Rab13 mRNA, and then shown that the Rab13 is indeed mislocalized when they treat the cells with these morpholinos. And then functionally, if we look at cell migration with the use of these oligos, we find that cell migration in 2D goes down. There's less invasion through matrigel. And also the collective kind of outward invasion of cells in matrigel in 3D is also decreased, all with this Rab13 morpholino. So there seems to be some functional significance of Rab13 localization in cells. Also, this is kind of all leading up to what we're going to be doing, but APC-dependent RNAs tend to associate with detyrosynated microtubules. So the Millie lab has also found that these detyrosynated microtubules kind of are associated with different types of RNAs. And on the left here, we kind of have this schematic, which is going to be important later, thinking about this cycle where tubulin tyrosynation and detyrosynation are kind of controlled, and the C-terminal tyrosine of the alpha tubulin can be removed by this tubulin carboxypeptidase, TCP, and then the tyrosine can be added back on by the action of the tubulin tyrosine ligase, the TTL. Okay, so later on, we're going to kind of perturb this cycle with different factors, but the presence of these detyrosynated tubulin or microtubules is controlled by this cycle. Also, it's been really interesting that the RNA localization also depends on the mechanical state of the cell, which the Millie lab has previously shown that the extracellular matrix stiffness can influence RNA localization. So in cells on very soft surfaces, there tends to be more kind of uniform distribution of the, this is a DDR2 RNA, as opposed to on stiff substrates, there's a more peripheral localization of the RNAs. And so we've kind of been thinking about this framework where mechanical tension and the cell's response to matrix stiffness and leading to the mechanical activity of the cell, how that can be kind of controlled through this pathway involving Rho A and ROC, which can ROC and MLCK can phosphorylate myosin light chain, leading to contractility, which kind of feeds back into this loop. But ultimately, this can also help promote detyrossinated microtubules and therefore RNA localization because they associate with the detyrossinated microtubules. So, so we kind of set out to explore different cell types. So let me just first introduce you to our metastatic melanoma cells. These are just A375 cells. These cells tend to rely on actin-myosin contractility. And if you treat them with blebbistatin in confinement, they decrease significantly in migration. And so they tend to utilize this, what we maybe would consider a traditional actin-myosin migration mechanism in confinement. In comparison, or in contrast, the MDA MB231 cells or S180 cells, these were the cell types I explored in the osmotic engine model of migration. These cells tend to rely less or can still migrate in the absence of actin-myosin contractility and maybe utilize more of this osmotic engine model when actin or myosin are inhibited. So these cells, the A375 cells and the MDA cells, we chose them because they seem to have distinct mechanisms for migrating in confined microchannels. And so we sought out to kind of answer this question of, does altered cell migratory behavior and the cytoskeletal dynamics in confinement affect RNA localization and or its function? So we started out with the question, does the cell peripherally enrich the APC-dependent Rab13 RNA? So again, we're going to be comparing both of these cell types, the A375 cells as a model of a mechanoactive cell and the MDA cells as a model of the less mechanoactive cells. And of course, we acknowledge that we have chosen two cell lines and we're kind of grouping them according to that classification, and we could do this on many more cell lines. That would be great, but we kind of stuck with these two to start with. And so then we kind of are working through this, these set of questions, which as I go through here, we'll answer them, but we'll talk about, is that peripheral Rab13 localization controlled by detyrossinated, the tubulin network? Is the peripheral localization and or the detyrossinated tubulin network controlled by the mechanical state of the cell? And does this localization actually contribute to confined migration? And again, we're comparing the A375 cells versus the MDA cells. So we first, to examine the effect of confinement on mRNA localization, we seeded the cells into these microchannels of two distinct widths, both 50 micron and then 3 micron wide. And these are all 10 microns high, just to note that. The 3 micron channel has a small enough cross-sectional area to induce nuclear deformation, which you can see here. And then we used fluorescence in situ hybridization, or FISH, to assess the distribution of different mRNAs. And in the paper, we looked at both APC-dependent mRNAs, but also a subsection of APC independent. I'm not talking about the APC independent ones today, just the APC dependent ones. So to quantify the RNA patterns, we measured that PDI, the peripheral distribution index, as I mentioned before. And the A375 cells displayed a peripheral APC dependent RNA enrichment in the 50 micron channels, which is consistent with the broadly observed localization of these RNAs and other cell types on 2D surfaces. And they maintained that same degree of peripheral APC dependent RNA enrichment in confinement. So essentially, there's no difference between the 2 microchannel widths for these two different RNAs, Rab13 and also KIF1C. KIF1C is just another gene that encodes for a fast plus and directed microtubule motor. The MDA cells, on the other hand, displayed peripheral RNA enrichment of both APC-dependent mRNAs. But then in the 3 micron channels, both of those RNAs had a more perinuclear distribution, so just a significantly lower PDI value. So again, there's this slight difference in confinement for the 2 cell types in terms of the RNA localization. Okay, so we have this first question: does the cell peripherally enrich the APC-dependent Rab13 RNA? So we have yes for the A375 cells and no for the MDA cells. So given this known involvement of the Rab13 mRNA in unconfined migration, we decided to focus in on that specifically. So the rest of the work here, we're just focusing on the Rab13 RNA. So our next question was whether peripheral Rab13 localization is controlled by that detyrosynated glue tubulin network. Again, these RNAs tend to associate with the detyrosynated microtubules. And so we hypothesized that differences in APC-dependent RNA distribution between the confined MDA cells and the A375 cells could possibly be driven by differences in the glue tubulin network. So we sought to assess different aspects of that glue tubulin network. So in the 50 micron wide channels, you can see the staining of glue tubulin, also just the overall tubulin levels. There was a detectable detyrosynated tubulin and alpha tubulin network in both cell types. So we have the MDA cells on the left and then the A375 cells on the right. This was consistent with peripheral RNA localization in both cases. In the 3 micron channels, the narrow channels, the glue tubulin network appeared reduced and fragmented in the MDA cells, which was in contrast to the A375 cells, which maintained a higher glue tubulin staining. I'll just note that the alpha tubulin network in the 3 micron channels appeared similar in both cell types. And then sorry, I keep going fast. We quantified this, and it was as we kind of expected, but the glue tubulin network appeared, let's see, it was, the signal showed that the MDA cells had lower glue tubulin signal than the A375 cells in both channel widths. So we kind of kept thinking about this, and we're thinking, how can we modulate this? And going back to this cycle of the detyrosynated microtubules. So we can modulate the levels of glue tubulin by manipulating the enzymes in this cycle. And so the tubulin carboxy peptidase, that's TCP, can be pharmacologically inhibited by parthenolide, which is a drug that leads to its inhibition. And we saw a reduction in the detyrosynated tubulin level, which we assessed with a Western blot. And so in the 50 micron channels, treatment with the parthenolide reduced the peripheral localization of the Rab13 RNA in the A375 cells. Again, this is consistent with prior reports on 2D substrates. And in the 3 micron channels, the parthenolide also reduced the peripheral Rab13 localization in the A375 cells. And we quantified this. Both were statistically significant when we looked at this over many cells. So the remaining glue tubulin network in the confined A375 cells seems to be functionally important to support peripheral RNA localization, since it decreased in both cases with the parthenolide. So we then did the same thing with the MDA cells. And we found that in the wide channels, treatment with parthenolide reduced the peripheral localization of Rab13 RNA, similar to the A375 cells. But in the 3 micron channels, the parthenolide did not affect the peripheral localization. And this was not statistically significant. So the fragmented network in the MDA cells doesn't seem to have a determining impact on the already less localized Rab13 RNA in the MDA cells. So then we wanted to test whether we could rescue the gluteubulin formation and consequently the RNA localization in the confined MDA cells. So to do this, we used siRNA mediated knockdown of the tubulin tyrosine ligase protein. So we used SITTL. And this indeed led to a significant increase in the Rab13 PDI, the peripheral distribution index in the confined MDA cells. So we were able to rescue this gluteubulin formation. And subsequently the localization of the Rab13. So kind of adding on here, the next question is peripheral Rab13 localization controlled by the detyrossinated gluteubulin network? It seems yes for the A375 cells, but no for the MDA cells. And so our next question was, is the peripheral Rab13 localization and or the detyrossinated gluteubulin network controlled by the mechanical state of the cell? Because remember that the A375 cells seem to be more or rely on a mechanism that's more mechanoactive in comparison with the MDA cells. So, and also this, I'll just remind you of the previous work from the Millie lab showing that substrate stiffness can impact RNA localization in cells, and it's kind of through this mechanical pathway in the cells. And so the first thing we did was look at YAP distribution in the cells, and specifically the cytoplasmic to nuclear signal ratio. YAP tends to translocate to the nucleus when cells are highly mechanoactive or undergoing mechanotransduction. So in the 50 micron channels, there was higher nuclear YAP in both cell lines. We had a YAP to cytoplasm to nuclear signal ratio close to 1. But then in the 3 micron channels, there was significant exclusion of YAP from the nucleus in both cell lines, but to a larger degree in the MDA cells. So the MDA cells had a significantly higher YAP, cytoplasm to nuclear signal ratio than the A375 cells. So this kind of supports the idea that the A375 cells still retain some mechanoactivity in confinement and to a larger degree than the MDA cells, perhaps. So we did look at the Actin network, and I don't have that here. It's in the paper if you're interested, but we didn't detect discernible differences in the Actin network between the cell lines in confinement. The Actin network appeared largely cortical for both cell types in confinement. So we decided to stain the cells with an antibody against active phosphorylated myosin. And when we did this in the 50 micron channels, we found that the MDA cells and the A375 cells had detectable active myosin. It concentrated in fibrillar structures, possibly reflecting actin filaments. In the 3 micron channels, the A375 cells maintained detectable active myosin, but the MDA cells tended to exhibit mostly diffuse staining in the bulk cytoplasm. There was some accumulation at the peripheral ends of the cells. Sorry, my mouse is very sensitive and I keep going forward. Just need to not touch it. So when we quantified this, we found that there was significantly more active myosin in the A375 cells in comparison with the MDA cells in both the 50 micron and 3 micron wide channels. So interestingly, the levels of active myosin, if you go back a couple slides, mirrored those of the gluteubulin, which highlights a potential connection between them. This was the same trend that we saw with the gluteubulin levels. So to explore that possibility, we used treatment with the myosin light chain kinase inhibitor ML7 to disrupt myosin activity. As expected, treatment with the ML7 led to a decrease in the phosphorylated myosin signal. And when we looked at the Rab13 RNA patterns in these cells, we saw that although the myosin activity was required for Rab13 RNA localization in unconfined cells, so we did see the drop in PDI here in the wide channels. Its loss was not sufficient to affect Rab13 RNA localization in cells in confinement. So these A375 cells, which are a little bit more mechanically active, if you inhibit myosin light chain kinase with the ML7, we don't see a difference in the PDI, the peripheral distribution of the Rab13 RNA. So something else is going on here. What other factors could this kind of driving this localization then? So we know that cells can respond to their external environment via the cytoskeleton, adhesion complexes, or stretch activated ion channels. And we were interested in this particular ion channel called piezo-1, which is highly expressed in A375 cells. So the Konstantopoulos lab has looked at piezo-1. They published a really nice paper kind of exploring this pathway in cells in confinement. But it stood out to us because A375 cells can maintain mechanoactivity through two independent factors, like these two pathways that seem to be redundant, which is the piezo-1 ion channels, but then also a signaling cascade that involves myosin-mediated contractility. And so we sought to look at the combined effect of piezo-1 activity and myosin contractility. So we found that knockdown of piezo-1 with two different siRNAs reduced the peripheral Rab13 localization. So you can see that in the images, the fish images, but then also quantitatively, both piezo siRNAs reduced Rab13 localization, peripheral localization in wide channels. With ML7 treatment, there was basically no change in peripheral distribution index. So there was no additive effect for cells in unconfined spaces with piezo-1 knockdown and ML7. However, we saw something different in the narrow channels. So in the narrow 3 micron channels, similar to the myosin inhibition, treatment with the piezo-1 siRNA alone did not substantially alter the Rab13 peripheral distribution index. So we saw that quantitatively here, there's no change in Rab13 PDI with either piezo-1 knockdown as with siRNA. However, co-treatment with piezo-1 siRNA and ML7 significantly decreased the Rab13 RNA PDI. So we saw that it was the combined treatment that actually reduced the peripheral distribution index. So these results support the idea that in a confined environment, the piezo-1 mechanosensitive ion channel and myosin activity seem to act redundantly to maintain peripheral RNA localization. And you need to target both of them in order to reduce that RNA localization.
Kimberly, sorry to interrupt you. I think if you would like to wrap up, because we have a bunch of questions you would like to address.
Absolutely.
Thank you.
Yeah.
Sorry about that. Yeah.
No, that's okay. I think it was just about done anyway. Yeah, so we looked at the role of the detyrosynated tubulin network, and I'll just summarize by saying that it was again the co-treatment that was needed to disrupt the glutubulin network. And so our last thing was, does this all matter? Is there a functional relevance? And so we used those morpholinos against Rab13 RNA and found that in the MDA cells, the Rab13 morpholinos led to a significant decrease in the wide channels, but there was no difference in the narrow channels. But in the A375 cells, the RNA localization actually did play a significant role for cells in confinement. So we kind of have this model where cells with a fibrillar glue-tubulin network, peripheral RNA localization that contributes to cell migration, whereas cells that have lower mechanical activity tend to have a fragmented glue-tubulin network, which leads to a lack of association with RNA localization, and then seems to not contribute to cell migration. So I'm going to leave it at that. And, have to share this slide with one of my PhD students put this together, all of the animals in our lab that like confined spaces as well. And then, of course, I need to thank all of the people, especially Dr. Becca Moriarty, who was the PhD student who did a lot of that work in the second half of the talk, Dr. Vula Millie, who helped as a collaborator with that project, and our funding sources. And again, thanks to all of you for your attention, and sorry for going too long. So I'm happy to take questions.
Thank you so much. No, you didn't go too long. We started late, and my apologies for that. That was 100% my fault. So we're just gonna lightning zoom through the questions. A bunch of them were from earlier in the talk. Perhaps you address things, but let's just go through them. So our first one is from Ekinola, who's asking about if you have any data where tumor cells and neutrophils were donor matched, and then could that possibly influence your transendothelial migration?
That's a great question. We haven't looked at that, and I'm sure there are. I'm sure you could do this with patients and get an IRB approval to probably do this. Maybe that is a really interesting direction to take. But we haven't done that.
Okay,
So very cool.
Awesome. Next question is from Prasoon, and I think there are a bunch of other questions which are kind of similar. Is asking the cells which were round in the shape of extra-linked stiffer material, where you were showing sort of the difference in this, I think the soft versus stiffness. The cells were elongated in shape. What was the reason for the opposite behavior? I think this is with respect to the hyaluronic acid one.
Yeah, like in comparison with PA gels, where we typically see cells more spread out on stiffer gels. I think that's what you're talking about, right? We don't know, but we would love to know. So yeah, we've seen conflicting results in literature too. We've seen at least one or two other papers that kind of corroborate what we saw, but then there was one other paper that kind of saw something opposite. So we're not quite sure. It could be the method of cross-linking. We used ethyl-based cross-linking, and other people have used methacrylate based cross-linking. So that could be a difference, which then leads to somehow changes the properties of the gel, so we haven't done any close examination of the like micro scale properties of these gels. I suspect there's some sort of interplay between the actual stiffness, but also other things that are affecting cell adhesion. That is my guess.
So I think Trevor had a similar question. I mean, you already said that, but Julie was asking whether AFM, did you like do any AFM measurements to verify the actual stiffness? Or
yeah, we did do that. Yeah. And they were in the range, like the single digit kilopascal range. We did do AFM. But again, I like with polyacrylamide, I guess there's some thoughts that you're changing porosity, which if you change the acrylamide concentration, you could be changing the porosity, which then can impact the amount of ECM protein that gets to the gel. So you do have that issue with the PA gels, but that might be even more of an issue with the HA gelatin gels. Like when you change the cross-linking, I think there's other properties like related to cell adhesion that are getting changed, that are affecting cell behavior. We're just not sure what it is. But it's definitely something we would be interested in trying to figure out. We didn't have the time at that point to do it. So
Thank you.
Yeah, no, I think, and there were, as Ankita said, there were multiple questions about that. And I was wondering about it too, because I think I've lied to my students, right? And stiffer substrates, bigger cells. And so
Yeah,
this
but
is really, that's really, really fascinating.
I think that's what we need to keep in mind. Yeah,
So,
absolutely.
I mean, maybe quickly, if I can add, I think the adhesion thing you're thinking might be absolutely true, because I was wondering whether the integrin signaling is much lower when you have this sort of, hyaluronic acid cross-linking jungle kind of a thing where cells can't really form focal adhesions and they have more rounded morphology.
Yeah.
Again, hand waving
it's super interesting, and lots of people here are curious about it, but we'll move on. The next question is from Nada, who is asking if you've noticed consistent patterns in the cell speed versus the microchannel size, and they're pointing out that the cells seem to be migrating faster with the with the smaller channel size from 6 down to and then the 3 micron channels seem to slow the cells down. And so if you could just comment on that.
Yeah, this definitely depends on the cell type. I've seen other cell types where like there's S180 cells. We've looked at actually in terms of cell division, the cell cycle, but those cells tend to decrease with all of the channel widths as we go down from 50 all the way down to 3. Others tend to be more biphasic. Like I think Nada's kind of indicating here with the down to 6 and then, increasing up to 6 and then going down up with 3. I think it just depends on the cell type. There could be a factor of cell size here too, because cells that are larger have a harder time squeezing into the really narrow channels. Nuclear stiffness could play a role. One thing we thought is that for cells in the wider channels, they have some degree, it's more like 2D migration, but then as you go down, the contact guidance can kind of guide the cells a little bit. So maybe speed goes up as you go with increasing confinement to a certain degree, like between the 6 and the 10 maybe. But then once you get into the more narrow channels, then the nucleus becomes a factor, and now that maybe slows the cell down. So this interplay between nuclear deformation and contact guidance could also be something that influences possibly or contributes to like a biphasic nature of the speed. But again, it's really dependent on cell type. And yeah, so there's not like a clear trend that's holds across all cell types that we've seen at least.
Thank you. I think we can ask Leonard to maybe ask his question since he has his hands raised. Leonard, do you want to ask your question? Yeah.
Yes, I do. Thank you for your great talk. Really, really interesting. I have a question about the osmotic engine model, if that's right. So on slide 20, you showed that if you change the density of the medium at one end of the cell to a hypotonic medium, the cell then changes direction. But I would kind of expect the opposite would happen, because hypotonic medium would result in water coming in as a result, right? Is that?
Yeah. Yep. This is a kind of a very difficult thing to explain, I think, actually, because it's kind of wrapped up in the mathematical model. So the cells can actually change migration even without, at least mathematically, without changing the polarization of the ion channels or the aquaporins. But experimentally, we see that after some amount of time, they actually do repolarize. And so you have this then like higher accumulation of the aquaporins at the new leading edge, which then allows the cell to start taking in water at that side, which then it can start migrating in the opposite direction. I'm not sure if that answers your question, but I think it's, yeah.
Yeah, more or less, I think. Sort of building on that, if I may ask one more further follow-up. I think in the paper you showed that, the aquaporins localized to the leading edge of the cell, but the model also says that water should go through the cell, so the cell doesn't necessarily like inflate forever, kind of. Mm-hmm. Did you look at other aquaporins to see if they localize to the end, or what do you think might be, like, how does the water go out, basically?
Yeah, that's a good question. We did not. So the cell types that we were looking at, I think aquaporin 1, 3, and 5 would have been good candidates. We focused on aquaporin 5, but absolutely, I think aquaporin 1 and 3 certainly could be playing a role here. And I think it would be interesting to try to target multiple isoforms at the same time to see what's going on. My lab is currently, so we have an NIH MIRA grant that's really focused on exploring how aquaporins are involved in different aspects of cell migration. So those are some of the questions we're really interested in, like these different isoforms. And is, if you knock down one isoform of aquaporin, is the cell compensating by overexpressing other isoforms? Or how is it using these kind of in tandem? So yeah, certainly they could have been acting too, but we didn't explore that.
Cool. Yeah, those are good questions to ask. So good luck.
Thank you.
Great. Thanks, Leon. Are you okay, Kim, to stay on for a bit? I know we're going a bit over time, but we have more questions. So a couple people, actually multiple people are asking about the role of the Rab13 RNA localization, and then some specific questions from Anna asking if the RNA is accumulated in granules, and then what is the function of the accumulation? Is it being translated at the leading edge? Is there correlation between the RNA and the protein distributions? Sort of just lots of questions on that.
Those are all excellent questions, and those are things we're trying to follow up with. Absolutely. Yeah, there's multiple ways that cells can get proteins to different parts of the cell, and I think transport, along microtubules, for example, is a major mechanism. But there's this kind of understudied question of whether the cells can localize RNAs to a specific location and then have local protein translation. So that's kind of something that's being explored. And I think something that we're definitely interested in. We didn't look at it in terms of this paper, but certainly a possibility. Yeah. And there's like lots of questions that stem from that. Are there other RNAs that are being localized, especially for cells in confinement? Yeah, and like kind of these questions about functionality. I mean, we showed that there was a very modest effect on cell migration, but what's the cell doing with these? Is there local protein translation? We don't know, but we are interested in all of those questions as well. So yeah, really good questions.
Great, thanks so much. Hani Suleiman is asking if you think the osmotic engine model it applies to non-transformed cells, for example, immune cells.
That's a good question. It's possible. And also I think there's a question of to what degree cells would use the osmotic engine model in vivo as well. We do know that aquaporins are involved with immune cell migration. That has been pretty well established. And one of the reasons why tumor cells are interesting is because they overexpress certain isoforms of aquaporins, and aquaporins are very much important for the osmotic engine model. So even if, and like kind of what my MIRA grant right now is exploring is these different ways that cells can use aquaporins, whether it's something like the osmotic engine model, or whether it's related to signaling within the cell, or whether it's pure volume regulation, or it has a lot of mechanisms that maybe through mechanotransduction, actually. There's a lot of ways that aquaporins are involved in tumor cell migration. And again, I mentioned that aquaporins are also involved with neutrophil migration. So it's possible that they might use something related to an osmotic engine model. But yeah, I'm not sure. We didn't explore tumor immune cells specifically.
Awesome, thank you. Next question is from Nile asking, it's from the slide 42 to 44, doesn't matter, but does ROCK inhibition also result in the same trend, since RNA localization seemed to be sort of downstream of Rho?
Yeah, that's a good question. We didn't inhibit ROCK. We just did the myosin-like minutes. Yeah. That's a good question. I think we would expect something similar. Yeah, but we didn't look at rock.
Great, thanks so much. We have a question from Mathide saying, thank you for the great talk. And then concerning the MDA cells on the gelatin HA gels, did you check the mechano responses of the cells on different stiffnesses other than spreading, such as focal adhesion, size number, nuclear localization, et cetera? So I think going back to that earlier, the earlier data, looking at some more of those typical mechanosensitive responses.
I'm trying to think. We didn't publish the focal adhesion data, and I'm trying to remember now. I need to look back at the paper, but I'm pretty sure that was not in there. And I think what we did have some of that data, but it seemed somewhat inconclusive. I don't think there were any noticeable changes. But I honestly, I would like to go back and look to see if that's true, because I may be remembering incorrectly there. But we definitely did not look at YAP nuclear localization. That would be actually a really good place to start, I think, here to try to tease out what's going on. That's a really good idea.
Right,
but I might
see
have
if it's what you'd expect.
Yeah,
cool.
Next question is from KSSP. I'm not sure who that is. Sorry about that. Is asking from the first part of your talk is availability of the migration track if the new set of cells was seeded on already degraded layer of HUX cells, or do they form some sort of a path or a void?
New set of cells. Sorry.
So I guess maybe let the cells get degraded and whole pores form in the HUVEX, and then take those cells off and put more on.
Oh, interesting. Yeah, no, we didn't do it. We didn't do it. The HUVEX were intact. They were kind of freshly formed into a monolayer, and then we added the tumor cells on top of that. But yeah, this is an interesting suggestion. We didn't try that.
Okay, has a second part also, which says for the A357 cells, do you see RAP13 RNA movement in live imaging? The sort of like.
Yeah. So we are currently developing the tools to be able to look at RAP13 RNA movement in live cells. Yeah. So we haven't done that yet, but we are developing the tools to do it. So that's a great question. Yeah.
It's always fun when people are like, have you done the thing?
Yeah,
and you're
right.
like, we're working on it. Yeah, so cool.
I think we have next one similar, Jen, I can take it, as like looking at the dynamics of ER and confined cells and how would that affect RNA localization, if you guys have done that.
Absolutely. That's a great question. We have not looked at that. So I can't answer that. Really good questions here.
I can actually copy paste it and like send it to you.
Yeah, right.
Yeah, that would be
great. My next grant proposal is already written for me here.
I think we actually, so we've already talked about ROC, so we'll skip that question. And then we have a last question about RAC GTP. And do you have any sense if RAC was different for the 3 micron pore size?
Another great question. We did not look at that though. Yeah, really good question.
Okay, great. Okay, so I think, if there are more last minute questions, folks, feel free to raise your hand or unmute yourself and ask. But otherwise, I think we will wrap it up. Thank you, Kim, for staying extra. Thank you, everybody, for your patience with our technical problems in the beginning. Next week will go smoother. So that was a really just tons of work and a fantastic sort of start to the cell migration season. So really excited to be able to have had you here, and excited to see what comes next over the coming weeks. So with that, I will end the live stream. So bye to everybody.